Greetings, this is Zahir Nali from the Solve MECFS initiative welcoming you to our third webinar of 2016. We are thrilled to have Dr. Jared Younger with us today, an expert in the biology of MECFS and explaining to us the tantalizing question, can we find answers in the brain? Dr. Younger is a director of neuroinflammation and pain and fatigue laboratory at the University of Alabama at Birmingham. He is also an associate professor there. Just a brief synopsis on Jared's background. He received his Ph.D. in Experimental Psychophysiology in 2003 at the University of Tennessee in Knoxville. He then completed a postdoctoral fellowship at the Arizona State University and then Stanford University School of Medicine before becoming an assistant professor at Stanford. In 2014, Dr. Younger joined the faculty at the University of Alabama in Birmingham with a primary appointment in the Department of Psychology and a secondary appointment in the Departments of Anesthesiology and Rheumatology. Professor Younger's goal is to end the chronic pain and fatigue that is caused by inflammation in the brain, especially in the context of MECFS. So we are really thrilled to have you, Jared, with us today. Dr. Younger is currently funded by the National Institute of Health, the Department of Defense, and several agencies as well. And what is really exciting today is Dr. Younger told me that he will be presenting a new information and new study designs that he has never presented before. So we are excited to learn about this as well. So Dr. Younger, without further ado, thank you so much for joining us and for doing this. The floor is yours.
Thank you very much. Let me make sure I have control over the slides. There we go. Okay, perfect. So I'm going to try to do this in 40 to 45 minutes and save some time for questions, but really what I want to talk about is one of the major goals of the laboratory, and that is how do we take an invisible condition like MECFS and how do we make that visible? What tests can we develop so that anybody can see what is wrong and they can clearly see it? So it's an objective test. So just like if somebody had a tumor in their brain, they could go to their oncologist, radiologist, they could do a brain scan, and then they could print that out and they could point to this part of the brain and say, there's the tumor, there's the problem, there's no doubt about it. That's the problem we have to take care of. So the question here is, what test can we do where we have that same ability in MECFS? You can print it out, you can look at it and say, here is the problem right here. So you've got to believe me because you can see it. It's obvious. Now, of course, the ultimate goal is to come up with treatments, and we spend a lot of time in this laboratory trying to develop new treatments. That's the end point. But the type of research I'm talking about today, which is to find that biomarker, that is a really important part of getting to the treatments, because if we don't know what to target, then we're just firing blindly, trying different treatments, and we waste a lot of money and effort. So we really need to determine where that problem is so we can have targeted treatments. So if we're able to come up with this objective test that anyone with chronic fatigue could take to see what's wrong, of course that would drastically increase credibility. So you would have much less dismissive individuals not believing in the condition. That would get a lot more researchers involved. Clinicians would be more likely to treat people with MECFS because they could see the problem, so it would get a lot more attention for the disorder. It would also really help with the research treatment because we could see what to target, we could track progress when we're trying a new drug. It allows us to come up with subgroups of people with fatigue, so it's really going to help if we can pull it off. Whoever can develop that objective test, that is going to significantly advance the research, but also significantly advance the treatment of MECFS. Now, there's a lot of places we could go to do that, so I'm sure people are doing blood draws. We do that as well, looking at things that are secreted in the blood, looking at the immune cells, looking at genetics. So there's a lot of possibilities, but what I'm going to talk about today is specifically the brain imaging, just looking at what happens above the neck. Can we find the signature there that's strong enough to actually be used for diagnosis and for treatment as well? So I'll show you some of the stuff. Now I do kind of a quick caveat, and this applies to anybody who's doing research in ME/CFS. One of the questions that come up a lot, people get really frustrated asking, why have we not figured out ME/CFS? Where's the progress? How come we haven't made these large advancements in this area? And I think there's a lot of reasons for that. You know, the funding needs to be increased. There's a lot of disagreement over the case definition criteria, so the people I enroll may be different from the type of people another laboratory enrolls, and that causes problems. And there's not enough researchers looking at this area, but those aren't the biggest problem. I think the biggest problem that we always have to keep in mind is that fatigue is not a disease, and it's not even a symptom. So fatigue is an alarm system, and it is the most general alarm system that our body has. So if you have profound fatigue day after day after day, month after month, year after year, that tells you something. It tells you that there's a problem somewhere in your body, somewhere in your brain, but it doesn't tell you where, and it doesn't tell you what systems are involved, because there are literally hundreds of things that could cause you to feel profound fatigue. I mean, just to mention a few, you could have, there's dozens of medications, there's thyroid endocrine problems, metabolic issues, you could have organ problems with liver or kidneys, there are multiple forms of anemia, multiple infections, viri, bacteria, vitamin deficiencies, other nutritional reactions. Could be a neurological problem, could be a reaction to an environmental trigger like spores or chemicals or even temperature, and that's just to mention just a handful of the many possibilities. So to rule out all of those things would cost hundreds of thousands of dollars for each patient, and you probably can't afford that, the doctor can't afford that, the researchers can't afford that. You would have to track everything you eat, everything you breathe to find the possible triggers. So that's kind of the problem, that with ME/CFS, there's probably people in there where there's actually different problems and we're giving it the same label. So when you hear someone talk about this is the biomarker for ME/CFS, I'm always skeptical when I hear that, because there's probably going to be more than one. There are probably going to be important subgroups. So just keep that in mind. It's just, as we're doing research, we have to realize that maybe we'll find something that explains half of them, but then we have to figure out the other half. And so we always keep that in mind as we're going through this. Let's see. There we go. Yeah, so I'm going to focus just on the brain. I love the neuroimaging stuff. That's one of the 3 techniques that we use. We also do clinical trials and we do immune testing, but the modern neuroimaging techniques, they're really amazing, and the advancements year after year are extremely impressive. I mean, we're getting great ability to see what the brain looks like. We get to see what's wrong with it, and there's a lot of techniques at our disposal. This right here, what you're looking at now, this is diffusion tensor imaging, which is you can do fiber tracking and you can look at the length and the connectivity of the axons that connect different parts of the brain, and so you can look to see if perhaps some of them are too short, or they're broken, or they're impaired, and so there may be inefficient communication between different parts of the brain. We're starting to do diffusion tensor imaging, but I won't be presenting right now, but basically this is an example of one of the techniques that we might use as we're trying to come up with a biomarker for ME/CFS. And really the goal here is not just to find something interesting or to find some place where the patients are different than the controls. We want something that is so strong and so obvious that you can clearly take that scan and say you have ME/CFS or you don't. And that's what we're aiming for. So one of the big questions is, have we accomplished that so far? Because there have been at least a dozen neuroimaging studies of ME/CFS over the past 15 years, and they use different techniques, but there's been quite a few. So have we accomplished that yet? And the short answer is absolutely not. In fact, we're not even close. If you look at all the literature, all the neuroimaging scans that have been done to date, and you take them all together, there's no overall story. This is my interpretation, but there's nothing you can pull out that gets repeated from all the studies that tell us actually anything that's wrong with ME/CFS. So Some studies will report that the gray matter of the brain is decreased in general, so that means there's fewer neurons, but other studies don't find that. They find that certain parts of the brain are smaller, but the region differs between different studies. One study will report it's the occipital lobe, which is kind of in the back of the brain. Another study will say it's the midbrain or brain stem that's kind of, I think I can point here. So this study found kind of the back of the brain was lower in ME/CFS. Another study found kind of the central area. Some studies will find that it's not the neurons, but it's the white matter tracts that I was showing with diffusion tensor imaging. So a recent Stanford study found increased strength in some of the axons. So this isn't a critique of any particular study. They may very well be correct. The problem is if we take the literature as a whole, we don't get a consistent story, and so we don't know what's true and what's not. So the critical thing with any neuroimaging study is to repeat it. You can find anything if you do it once, but if you do a study and you get a result and then you do it again with a new group and you find the exact same thing, then you're on to something. So, you know, I'm really encouraging everyone to try to find the money to repeat the studies to see if they have something that's kind of robust. But this isn't a review of the past literature, so I'm doing one slide on that just to show people that we've got a lot of work to do as we're developing a biomarker. Oh, this is, I tell you what, I'm going to do one little extra caveat for neuroimaging, and then we're going to launch into the good stuff. You might have seen this before. This was a Dartmouth study by the psychology department where they took a dead fish, I think it was a salmon, they put the dead fish in the MRI scanner, and then they showed the fish emotional videos, and they found that the fish brain reacted to the emotional videos, which is, of course, is ridiculous. And what they were demonstrating here is that if you don't do your statistics right, if you have your threshold too low, you can pick up noise and it actually looks like signal. So you would conclude that dead fish brains react to emotional videos, which is kind of crazy, but that's what they found. And it was a warning to neuroimaging researchers to be careful with their statistics because not all the people who publish are very careful with the statistics. I'm going to cover this in 1 minute and then we'll get to the cool results. But just to kind of demonstrate that point, because I want people to be skeptical reviewers of neuroimaging research because you can't just automatically trust stuff just because it comes up in the brain. Again, you have to repeat it. This right here is from a study I did where I gave people opioid painkillers and we wanted to know if it changed the brain. Well, depending on where I set that statistical threshold, I get really different results. So if I set it really high so I can be really confident in the results, nothing survives. So you can see that there were no activations. They would come up as color clusters. There's nothing here. So if I set the statistical threshold really high, I would conclude that opioids don't change the brain. But if I relax that threshold a little bit, you start to see places where opioids do change the brain. And so then I would conclude that opioids change the brain a little bit. And then if I relax that a little bit more, and these are all perfectly acceptable in the literature, so I could have published any of these. Now I find that opioids change the brain a lot. So just by changing that statistical threshold, I could have published a study saying opioids don't change the brain. I could have published a study saying they do. And that's a little scary, but that's kind of how it works. So this isn't necessarily a huge problem as long as the researchers are paying attention to what they're doing and openly reporting what they're doing. So I don't want to say that this is all nonsense. The neuroimaging techniques are really, really powerful, including the ones I'm about to talk about. But again, the key thing is we have to see it repeated more than once before we can trust it. So that was just a little caveats, and now let's talk about some exciting things. So most of the things I showed you previously were MRI, that's magnetic resonance imaging. MRI does some really cool things. It allows you to see the shape of the brain, allows you to see blood flow, but it's tough to see what's actually happening with MRI. So we really need other techniques if you want to find the mechanism of ME/CFS pathology, what's actually driving the problems. And PET, positron emission tomography, that's one technique I think is going to be really important as we try to come up with a biomarker for ME/CFS. And I really like PET because it allows us to tag something and then we can see where it goes in the body. So if I wanted to know where dopamine is in your brain, we could take dopamine, we could tag it with this radio tracer, inject it in you, and then we can do a brain scan and find out where it's happening, where it's showing up, and we know that's where dopamine is going. So there's a lot of flexibility. We can explore some really interesting systems. Now this is a radioisotope, so it is radioactive to a low degree. It releases photons that we can pick up with the scanner, and so while it's a very powerful technique, and I'm going to show you we can do some really potentially interesting things with it, there are some downsides. It's very expensive. It costs about $5,000 per scan, so you have to know what you're doing before you start a study because you run out of money really quickly. And because you're injecting a radioisotope, it's not something you can do a lot. You could do it 2 or 3 times a year to be safe. Whereas with MRI, you could go in every day if you wanted to, and there wouldn't be a problem. So what can we do with PET, and can we actually figure out ME/CFS using PET? So I'm going to show you a study that another group did, and then later I'm going to tell you about what we're going to try to do with PET. But the reason why PET may be very interesting is, you might have seen this slide if you've seen a talk of mine before. These are microglia cells, and our lab hypothesis is that abnormal microglia cells are driving the symptoms of chronic fatigue syndrome and ME. So this is a normal microglia cell, and it has these little branches that are looking for problems. These are only in the brain. Now when they find a problem, they activate, which you can see here in the lower right. When they activate, they pull in their arms and they start to pump out a lot of pro-inflammatory chemicals, which is exactly what happens when you get the flu. And that's why you feel so bad when you're sick, because all these pro-inflammatory chemicals come out and they interact with places in your brain like the amygdala, the anterior cingulate cortex, the hippocampus, the hypothalamus, and when you have activity in those regions, in those particular subregions, it makes you feel ill. It basically gives you a feeling of malaise, of fatigue, and so when someone gets the flu and they can't get off the couch, these cells are what are responsible, and the chemicals they produce and the way it changes the way your neurons function. So this is the source of feeling sick. This up in the upper right, this is a cell that's kind of aggravated, what we call primed, and you can see it's got more feelers and there's more receptors. And so we think in ME/CFS, for some reason the glia, because they've been exposed to something, we don't know what that something is, but the glia are now kind of angry, aggravated glia, and now it takes very little to set them off. So now something just normal, just taking a walk, may cause these things to jump into the activated stage and make you feel horrible. So that's the basic idea. Now you could never look at this with MRI because these things are way too small, and MRI is just, we don't have the resolution to see. You can't image these cells in a living human. You can only look at these by taking the brain tissue out and examining under really powerful microscopes. But with PET, we can possibly investigate it. So this was a 2014 study, pretty recent, by Nakatomi and colleagues, and they actually, to my knowledge, this is the only PET study in ME/CFS. I might be wrong, but it's the only one I know of. And they injected a tracer that goes to a receptor on activated microglia. So it's not too responsive to normal microglia, but it goes to activated microglia. So we kind of consider it a marker of microglia activation. And any place where you have activated microglia, you're going to have the release of those pro-inflammatory chemicals, and that may be making you feel sick. So I really like this study design. So they inject this in the people, then they scan to see, okay, is there any place where the ME/CFS brain takes up more of this chemical than the healthy brains? And that would suggest that in the patients there's more activated microglia, and that may be what's wrong. And they did indeed find that. They found a lot of regions of the brain where the patients were taking up more of this chemical, suggesting that the microglia are abnormally activated. And this is kind of the main results down here. You can see that it's always the case that patients had greater uptake than controls, which is what we would hypothesize. In some cases, like the cingulate, which is a kind of an emotional processing region of the brain, there was 200% more uptake of that chemical in the patients than in the controls, and that's really, really powerful. What I like about this study is all the regions make a lot of sense. These are all regions that are associated with your mood, with fatigue, with your energy. The thalamus is really important because it's the switchboard of your brain. Amygdala controls your motivation, so if you have problems in that area, you're going to have some depressed mood. It's going to be hard to get motivated. So a lot of these things are associated with different forms of fatigue. And so it's exciting in a way from a scientific perspective to see so many of these come up in this scan. Now, again, to my knowledge, this has only been done once. This is very potentially exciting, but they have to do it again, or someone has to do it again to make sure it's true. But if this is true, this is a pretty major advance, because this isn't just saying, hey, the patients are different controls. This is actually saying why. So if this is true, this indicates a particular line of treatment. We can give drugs that can get to these areas and calm down those microglia, and that may help ME/CFS. Now, there's one other thing with the study that I really liked, and because I was telling you that we need scans that can clearly separate patients from controls. So in this thalamus region, which is very, very important, look at this right here. These are the healthy controls and how much of that chemical they took up, and here's the patients. And what's really interesting here is there's a clear division. All the patients had higher levels than the controls. That's what we need in a diagnostic test. We need an absolute separation. So you can say, you know, if you've got uptake of more than 0.22, you're probably going to be a patient. This is the separation that we need to really make an advance. And so I'm really glad they found this. Again, I hope that someone can repeat it, but preliminarily, this is very, very interesting. Let's see. I think there's something else I want to say. Yeah, in this thalamus area, the reason why I find that so interesting is, you know, you kind of think of the thalamus as the dispatcher for 911 for emergency services. So you call into 911 if you need help immediately, and you call the dispatch office, and that operator will listen to you, decide whether to call the fire department or police. This is like getting that operator drunk, and so your house is on fire, you call in 911, and instead of them sending the fire department, they send the police department or something. So the whole system breaks down if that operator is not doing their job. That's what the thalamus is doing. It's controlling and coordinating all the areas of the brain. So if you have disruption or inflammation in that area, that can cause very widespread problems with how you feel, how you think, your fatigue levels, motivation. So this makes a lot of sense, and I hope it's something that repeats, and that'll be really interesting. So I want to tell you about the things that we're going to try and how we're trying to get a improved scan for ME/CFS. I'm going to talk about two things. One of them I've talked about very, very little. The other one, as was mentioned previously, I've never talked about before outside of our research group. So I'm going to take a kind of a risk here and get the news out before we've even run the study. You know, scientists usually keep things pretty secret because we don't want anyone else to copy our work or beat us to it. So usually you only hear about things after the study's done, but I'm going to try this experimental thing and tell everyone what we're doing before we do it, and we'll keep doing that as long as there's not a problem. If someone steals one of our ideas, maybe we'll change that in the future, but for now we'll talk about it and be open with it. So my goal here, as I mentioned before, is I want to scan where if I took 50 results and half of them were patients and half of them controls, I could look at those scans, I could put them in two stacks, one for patient, one for healthy control, and be 100% accurate without looking at anything else, without knowing anything about the people, just by looking at the scans. That's the goal. So the two things we're going to try to accomplish that, one is brain temperature, and the other one is immune cell tracking. So I'm going to talk about the brain temperature first. This is a pretty interesting thing. It's pretty new. I've talked about this, I think, once before. I'm not sure I've ever presented to a large audience, but I think on our YouTube channel I did like a 5-minute video on this. So we're still in the very early stages of this, but so far it's actually looking pretty interesting. So let me give you the basic idea of how this works. What we think will happen is when there's neuroinflammation in your brain, and we think that the neuroinflammation is driving the symptoms, we believe there'll be an increase of brain temperature. Just like in your body, when you have the flu or a bacterial infection, your body temperature will go up. We think when there's neuroinflammation, your brain temperature will go up. So we use temperature all the time as a marker for illness. You know, if you think you're sick, you'll put the probe in your mouth, you'll measure your core temperature, and you know if you have a fever, you're probably sick with something. Your immune system is activated. So we're just taking that concept and we're moving it to the brain. So the idea here is that if the immune system in the brain gets activated, it's going to generate extra heat, and the blood coming to the brain from the body will not be sufficient to cool the brain. So it's going to continue to warm up. And the key thing here is that you may be able to have elevated brain temperature but normal body temperature. So the important thing here is just taking your body temperature, putting that thermometer in your mouth, you won't see the problem. You have to measure the brain temperature specifically. Now that's complex to do. You can't do that with a thermometer because you can't get to the brain. And so we have to find some other way to do it. And the way we do it is with a modification of magnetic resonance imaging, and that's called magnetic resonance spectroscopy. So instead of looking at a picture of the brain, we can actually look at chemicals. So let me show you how that works. So this is an example of a MRS, magnetic resonance spectroscopy scan, and you can see here it's looking at this particular area of the brain. So this brain, we're looking from the top down, it's kind of in the middle part, and we've drawn a square over this, and then we can look at the chemicals. So MRS gives us concentrations of different chemicals, a very different technique in MRI. So we can look at some different chemicals. The interesting thing is chemicals live on a particular frequency in this axis here. So NAA, which is N-acetyl aspartate, always lives right here. So you can always find it in the same spot. So where the peak lies in this direction tells you what it is, and the height of the peak tells you how much there is. And the reason the NAA peak is so big is because it's a marker of neurons, and of course your brain is filled with neurons, so there's going to be more of that than all these other things. So very, very useful for quantifying what's in that region of the brain. And you can look at many other things. Some of them are interesting, some less interesting. By the way, one other study, I think it was 2012, they found increased lactate in the brain, which is very interesting because lactate is produced when you run out of oxygen in that region, and that suggests that there's very profound neuroinflammation occurring there. So that's another interesting way to use MRS, but that's not what we're using it for. So we can look at all these chemicals. Now some people may be wondering, okay, what happens if you keep going? What happens after 4? And most people don't look over there because they're not interested in what's over there. If you go past 4, you see this huge colossal peak that's water. That's because we're mostly made up of water, so that's not surprising. It's much larger than anything else you'll find because of the concentration of water in the brain. Now again, no one cares about water. You know, it's there. It's not the cause of ME/CFS, so no one researches it. But water has a really interesting property that we can use to determine temperature, and that is almost all of the peaks are not affected by temperature. So NA, for example, is always in the same place no matter what the temperature of that area is. And that's the same with these other peaks, except for water. Water moves right and left depending on the temperature. The hotter that area gets, the more the water peak moves to the right. So what that means is if we find the NAA peak and then we find the water peak, we can calculate the distance and we can determine the temperature. We just have to solve an equation. And so the closer the two peaks are, the hotter that region is, the further away they are, the cooler that region is. So we can do that in the brain and we can determine the temperature of that exact spot we're measuring. And we can do that without touching the brain, just putting someone in the MRI scanner. Nothing is injected, there's no probes, you just lay in there. You could do it very quickly and we can get temperature. So very, very cool, very exciting technique. Let's see. Yeah, and then we can combine this with a technique that allows us to look at this in multiple voxels. And so a voxel is just a square in the brain, and we can do this not just in one place, but all over the brain. And you can see that the results are a little bit different in different parts of the brain. And so we can get a temperature measurement in each one of these areas. I think I got a spot. Yeah, so these are in Celsius. I'll convert some of these to Fahrenheit in a little bit, but we typically read these out in Celsius. These are pretty normal brain temperature. You know, they're around 99 degrees Fahrenheit. So what's really interesting is we can get the whole brain. I'm just showing you a little square in the middle, but we can do this all the way out to the sides. We can go front, back, and we can go all the way from the top to the bottom and get measurements in every little square. So we'll get thousands of temperature measurements in one brain. Now we can use that in a couple of different ways. We could average those and get your overall brain temperature. That's a very useful thing. But we can also do temperature maps. This may be really, really interesting because we can color code these things where green is normal temperature, yellow is slightly elevated temperature, and red is significantly like fever region temperature, like over 101 degrees Fahrenheit. And so these are really neat. If we do this across the entire brain, we can look for hot spots. And it may be that those hot spots indicate regions where there is acute inflammation, and that could be very informative. So remember I showed you earlier those people who did a PET found that area of the thalamus, it was particularly the left thalamus, where they're saying there's neuroinflammation. We may be able to do this temperature scan and find that that same region of the thalamus is actually hotter than the rest of the brain, suggesting that there's neuroinflammation. The reason why you'd want to do temperature is because this is significantly cheaper than PET, and it's safer. So you know, we can do this for probably $250 as opposed to $5,000 per scan, and you could do the math on that. If you're running 10 or 20 people, that's an enormous cost difference. So this is a much cheaper thing, and it's actually easier to do. So once we figure this out, a lot of other groups can do this as well. So that's the temperature. Now let me show you our very first results. I've only talked about this once. We've only tried it on a few people because we're basically trying to optimize. We're trying to get the whole brain as fast as possible and as accurate as possible. So we're still optimizing things. We'll probably start running the studies in about a month to a month and a half. So those are going to be starting up soon. We're almost there, and things are looking pretty good. But we did get about 5 people to do the scan and look over their brain. And so I want to show you what this has done so far. So we have 2 healthy controls, and then we have 3 people with CFS. 2 of them just have straight chronic fatigue syndrome. 1 of them had chronic fatigue syndrome and also had rheumatoid arthritis. And we've also looked at some straight rheumatoid arthritis individuals, and I'll show that another time. So this is overall brain temperature, and again this is Celsius. I'll convert it to Fahrenheit. So the healthy people have basically the same brain temperatures, and that's about 99.5 Fahrenheit. So that's not what we would consider a fever. The brain is going to be a little bit hotter than the body because the brain's kind of locked inside the skull, and so there's not as much surface area to dissipate the heat. So it's going to run a little warmer in everyone, but that's still a very normal temperature there. The 2 CFS people, though, again, almost exactly the same brain temperature between these 2 individuals, and that's 100.5 degrees Fahrenheit. Now, we usually consider a temperature of 100.4 to be kind of a fever, and so these individuals had elevated brain temperature that we would consider to be kind of in the range of fever, and so this is definitely hot enough where it could be driving their symptoms. And then the person with CFS and rheumatoid arthritis had an even higher temperature of 101 degrees Fahrenheit, and that's you know, something that definitely could be driving the symptoms. So just in the few people, there's clearly the people with chronic fatigue have higher brain temperatures than the healthy controls. So far it's looking good. Another thing to note, all of these people had absolutely normal body temperature, so no one had a fever in their body. So again, just looking at their body temperature wouldn't have told you anything about what's going on. So as far as how we're going to use this, we have a funding pilot grant from the Rheumatology Research Foundation to look at this in rheumatoid arthritis, and the reason we're doing that is because a lot of people with RA will get treatments and it'll help their joints, but they still have profound fatigue, and we believe that's because the medicines help joint inflammation, but they don't cross the brain to help brain inflammation, and so they'll still stay fatigued. So we're going to do that, but we're also going to look at, we're trying to get funding to look at chronic fatigue syndrome, fibromyalgia. We're also going to look at traumatic brain injury, and we're going to look at multiple sclerosis. So we're going to be doing a lot of these scans starting this summer. And we're optimizing the parameters. So we're going to see a lot more of this. As soon as we get some results, we'll put those out, and discuss, and then we'll see if this could potentially be a biomarker where you could just go in, do the scan, and if you have a brain temperature of over 100.4 degrees Fahrenheit, that may suggest you have neuroinflammation, and that would guide some treatment decisions. So the other one, this is the brand new stuff that hasn't been discussed before, and I'm really excited about this one because this is something that, if it works, it will profoundly change how we think about ME/CFS, and it'll change how we treat it, and it'll open up a lot of interesting directions. So here's the basic idea. We have a peripheral immune system and we have a central immune system. So in the brain, the immune cells are called microglia, and they're responsible for the whole immune defense of the brain. In our body, we have T cells, B cells, natural killer cells. There's a lot of different cells in the body. Now those are supposed to be kept separate. So your peripheral immune cells, your T cells and B cells, they're not supposed to be in your brain, and your microglia in your brain are not supposed to be in your body. Now the blood-brain barrier, which is a kind of a sheath that goes around all the vessels in your brain, it keeps those two things separate. It keeps the peripheral immune cells out of the brain because those peripheral immune cells are not trained to operate in the very sensitive environment of the brain. That's potentially dangerous if they're in there for too long, if they infiltrate the brain. So the blood-brain barrier does a really good job of keeping those out. There's a couple different layers. There's these little tight endothelial cell junctions, and then the astrocytes, which is some cells in the brain, they form these little lipid feet that form a different barrier. So there's a couple of barriers you have to get through to penetrate into the brain. And in a healthy individual, these things should always be kept separate. Now what we're hypothesizing, what's possibly happening in ME/CFS, is something has happened to this blood-brain barrier. Either it's been damaged, or for some reason the astrocytes are kind of opening the gate. And what is happening, again, this is just, we haven't shown this, this is just a hypothesis, is that these gates have opened and it's letting the peripheral immune cells squeeze through. They squeeze through into the brain, and then they start causing inflammation, which is making you feel fatigued and making you feel sick. Now this picture right here is from multiple sclerosis, and we think this also happens with multiple sclerosis, but in MS, the cells that get through are trained to attack your own neurons, and so that's a very devastating illness. In ME/CFS, we don't think autoimmune cells are getting through. We think other cells are getting through, but they're enough to cause inflammation. They're not directly attacking your neurons, but it's creating inflammation that's making you feel sick. So kind of a lesser form of multiple sclerosis would be one way to look at it. Not as damaging, but still makes you feel really bad. So if that were to happen, all these cells in the periphery are going to punch through and start to cause inflammation. And there's many cell types. You got your B cells, your NK cells, your T cells, and these could be causing an inflammatory response in the brain. So I think this is why you've probably saw recently that rituximab, which is a B cell depleter, why it may work in ME/CFS, because if you can give rituximab and deplete the B cells in your body, there won't be as many B cells to cross into the brain to cause problems. That's just my guess as to how it works, but it all fits together with the hypothesis. So anyway, how do we do a brain scan to see if this is happening? Because if we can find these peripheral immune cells in the brains of people with ME/CFS, that's going to be a breakthrough because they're not supposed to be there. So that would be something where you would say, oh look, here's the immune cells, this is probably what's causing the problem. They're not supposed to be there. So to do that, we have to go back to positron emission tomography. We cannot do this with MRI right now. We may be able to in the future. We're looking at that because it's cheaper, but right now we're going to use PET. So what we do here is we take a person with ME/CFS or a healthy control and we do a blood draw. And we take that blood and we separate out the leukocytes. And the leukocytes are basically all these cells, all these immune cells I was talking about. Then you take those immune cells and you incubate them with the radiotracer. And so those immune cells will eat the radiotracer, and so now they've got the tracer inside them, and then we can measure where those cells go. So once they've been with that tracer for about an hour, they eat it up, the cells are tagged, now we inject the person's immune cells back into their body, except now they're regular immune cells, they still work, they just have a tracer inside them now. So now we can follow them. Now, what we'll do is we'll inject these cells back, and then we'll bring the person back 24 hours later to scan their brain to see if any of those cells actually punch through into the brain. And then we'll bring them back about 3 days later to see what it looks like. What we expect to see is that in a healthy brain, no signal will be detected at all. There'll be no evidence of infiltration of the brain. But we think in the ME/CFS individuals, we'll pick up that signal in the brain. Now, what's really cool about the scan is that there's no background signal. So if the cells don't get to the brain, there will be nothing there. It'll just be total black. Other scans, there's a lot of noise. There's no chance of noise. So if there's no cells there, you're just going to get a black picture. Very easy to interpret. If there are cells there, you'll get a signal. It'll be kind of glowing on the screen. And so you can say yes versus no. It's as simple as that. Yes, the immune cells made it to the brain versus no, they didn't. So if we find consistently that the ME/CFS individuals have these cells in the brain but the healthy controls don't, that's going to be a major advance, because then we know we can target central inflammation, or we may need to target those immune cells in the periphery before they have a chance to cross the blood-brain barrier, or maybe we need to try to give agents to repair the blood-brain barrier. It opens up a lot of targets for treatment. So we hope that works. We don't know. We have not even run the first participant. This is very, very complex stuff. Probably take us about 5 to 6 months before we're truly running participants. I'm working with Suzanne Lappy, who's an excellent radiochemist here at UAB. So we're going to get there, but it'll be probably later this year before we're actually running this full steam. But you know, it's the kind of thing where, again, if the hypothesis is supported, it's going to be very clear to interpret, and it's going to really change how we approach CFS. So that's kind of a taste of what we're going to be working on, and the promise, I think, that neuroimaging has. Really, the question is there's so many neuroimaging techniques. As researchers, we have to figure out what are the ones that's going to give the clearest answer, which scans are actually going to get us closer to a treatment. We need to make sure we're running the right scans. So, you know, the way I see this in my job, as I'm trying to decide what I do day after day after day, the way I think about this is, you know, in the United States there is at least 1 million people with ME/CFS. That might be conservative, but let's just say it's 1 million people. That means every year we lose 1 million years of productivity. So it's just, if you think of it in aggregate, it is a colossal waste of productivity and quality of life to have that many people suffer for that long. So if it takes us 10 years to come up with a treatment, that's 10 million years of lost productivity, and that should be absolutely inexcusable. So the key here is we've got to move fast, and it's time for us to do really high-risk studies that might not work, but if they do, it's going to answer a fundamental question, and it's actually going to progress us. So in our lab, we're asking the very big questions. We're doing very complex studies. They're hard to do, but if they work, that's a significant advance. And I encourage everyone who's interested in ME/CFS to kind of think that way. We've got to move fast, and we've got to try things that are risky. I don't mean from a health perspective, just they're risky in that we don't know whether they're going to work or not. That's what we're doing from the research side. Now, as far as what patients and concerned individuals can do in the meantime, there's a few things I want to throw out to you if you want to keep up with the science. There are excellent blogs, and Solve ME/CFS does a great job at disseminating this information. Health Rising, Phoenix Rising, and there's really a lot of good solid sources of information, so you probably know about those. But a few other things I want to throw out to you. One is an application that a friend of mine from Stanford wrote. It's called a Stork app, and that's storkapp.me, and you can go there, put in your email address, and then you type in keywords like chronic fatigue or myalgic encephalomyelitis or fibromyalgia, and every day it'll send you an email after it looks through the scientific literature, and it tells you if new papers came out. So you can look at abstracts of all the new results every day, and so then you find out stuff as soon as the researchers find out. Also looks at grants. So if someone in your state just got a grant to test a new drug for ME/CFS, you'll find out about it the same day it's awarded. So that's a pretty cool app, and I use it and I read it every day, so I stay on top of the literature, but it's actually available to everyone. Another place to go is clinicaltrials.gov, because any clinical trial that's done, we have to register it, and so go there, type in chronic fatigue syndrome. Unfortunately, right now, there is not a lot of clinical trials, if any. We're going to be starting some in the next month and a half, but you can go there periodically and see if there's any clinical trials. We've got our lab, we have our YouTube channel, and I try to put out videos every week or every 2 weeks so you can stay on top of what we're doing. And again, there's blogs. So just kind of as a final note, I've said this before in a couple places, but I do truly believe that, you know, kind of the ray of hope is I am definitely, without a doubt, seeing increased attention and awareness and funding for ME/CFS and those associated disorders, and that's the result of a lot of people making it a priority to change the way that the federal government looks at this and to raise awareness for the issue. I can tell you that those efforts are working, and I can see the shift that's occurring that's going to lead to advancements more quickly. I see the difference in the NIH. They're taking this very seriously. I see it in the CDC. Things are actually changing. So you know, I really appreciate the people who have made those things change, made those things happen. I know the results and the treatments can't come fast enough, but there really are promising things on the horizon, and I truly am optimistic in that there'll be some major advances pretty soon. So we'll work on that side. In the meantime, you know, keep an eye out on the new science. There's a lot of good places to see when new information comes out, and hopefully one of these brain scans I talked about will work, and within a year we can present not just exciting ideas, but we can actually present some exciting results that can actually lead to treatments. So that's an overview about what we're going to be doing in the next year as it pertains to trying to find a biomarker for ME/CFS. And that's what I have. Thanks.
Great, fascinating. Thank you, Dr. Younger. This is wonderful. You took us on a journey from the diversity of the signals to the complexity and symptoms of ME/CFS, to the statistical analysis, to neuroimaging, to the glia biology, to thermometry, to immune cell tracking and peripheral immune cell tracking. All of these are fascinating. Let me just sneak in one of my questions before going to our guests' questions, our audience. You mentioned glia, primed glia, as a potential contributor. Now, can you elaborate a little bit on how you modulate these glia, or how you down-modulate their effect back to normalcy, and whether there are drugs available for that? You kind of touched on this very briefly.
Yeah, these microglia cells, they're amazing. They can snap in and out of that activated state in minutes, if not seconds. So I've seen videos where these things can go from normal to activated in less than 30 seconds, and they can go back. So there's multiple states. There's actually more than what I talked about. There's the normal state, there's this kind of aggravated, angry state, then there's that activated pro-inflammatory state. There's also another one which we call the alternative activation state that's actually neuroprotective and calming. And so exactly what you're saying, we're trying, and other groups are trying to work on drugs that can coax those cells into the state where we want them to go. Now, there's some things that are already available, at least to researchers. One of those, you probably heard me talk about low-dose naltrexone. When you block a receptor called toll-like receptor 4 on microglia, they tend to go back into their resting state. So that kind of calms them down. Minocycline does that, dextromethorphan. So those are some pharmaceuticals. Ibudilast is another one. And I can't recommend these as treatments because they're all kind of in the research stage. But there are multiple drugs that do what you're asking. It modulates the microglia. And it also looks like there's a lot of kind of supplements and herbs that do this. So curcumin, which comes from turmeric root, resveratrol, trying to think of a few other ones, French maritime pine bark, and there's probably about 20 that have been demonstrated in animal research to push these cells into a calm position. So exactly, that's kind of the short answer, but Yes, there are lots of agents, and that's a huge field now. What drug can cross into the brain and get the microglia to go into the position we want them to be in?
Great, great, thank you. So here's a question from our audience and listeners. Can you explain the blood-brain barrier in novice terms?
Yeah, it's basically just a wall that's made of cells, and they only allow certain things to pass. So to pass through the blood-brain barrier, it has to be a very, very small thing. So most things are too big. The blood-brain barrier keeps out almost everything, so very few things can get through. That includes about 98% of all drugs that could help in the brain, but they can't make it. So when you take a drug, chances are your blood-brain barrier is going to keep that out. So you think of it as a wall, but it's a wall that has gates, so they can be opened, and it's almost like these cells in the brain are guards, and if they see something they want to get through, they can temporarily open the gates and let that thing through. For example, if you have a horrible neuroinflammatory disorder and the immune cells in the brain, I want to say think, I mean they're not actually thinking, but if they decide that they need help, they'll open up those gates and let in these peripheral immune cells to kind of help whatever that infection is. That can cause more problems in the long term, but that's kind of how it works. So it's basically just kind of a wall with gates, and it doesn't surround the brain. A lot of people think of the blood-brain barrier as surrounding the brain in total. Actually, it surrounds the vessels. And so this wall is all throughout your brain, on the outside, inside. Any place where there's a blood vessel, your blood-brain barrier is surrounding that to keep out these chemicals from the brain.
Great, thank you. Another question from our listeners: how does brain inflammation cause body fatigue? That's a fundamental question too. How does brain inflammation cause body fatigue, and how the two are related?
So, the important thing to understand with this one is when we think of body fatigue, you think that the fatigue is actually in your body because that's where you feel it, but that is a complete illusion. Everything you feel in your body, you're actually feeling it in your brain. The fact that it feels like it's in your body, that's an illusion being created by your brain. You can't actually feel pain in your body, which is kind of crazy to think about. But if you take a pen and you poke your finger, you feel it in your finger, but that's like a movie. It's fake. Your brain is tricking you into thinking you can feel that in your body. That sensation is being created by the somatosensory cortex in your brain. It's the same thing with fatigue. All fatigue is in the brain, no matter what it feels like. You can't feel stuff in your body. All your body does is collect signals and send it. So your brain has maps of your body, and when you feel pain in a certain region, there'll be activation of those neurons, and you interpret that as feeling something in your body. So that's the way you can feel fatigue in your body, is that it's still a brain disorder, but it's being projected into the map of your body. So the reason why brain inflammation can lead to fatigue is because if you have these pro-inflammatory factors, when they go to places like your amygdala and they go to a place called the anterior cingulate cortex, and they change the way the neurons work in those regions, that will cause you to feel fatigue throughout your body. And I know there's different types of fatigue, but it'll cause body fatigue, probably also mental fatigue. So that's kind of how it works. It's because everything actually happens in the brain no matter what it feels like to you, and these pro-inflammatory factors produced by the microglia change the way those neurons operate so that you actually feel malaise, sick, fatigued, maybe depressed or slightly depressed, maybe anxious sometimes, and all that happens in the brain.
This is great, and there is a related question here. I'm just gonna, you answered it already. This is a general question. I find it fascinating, and the explanation you gave is so on point. This is the question. This is fascinating and exciting stuff. We agree, but why would brain inflammation cause problems in ME/CFS patients often have elsewhere, like muscle aches, constipation, food intolerance, and acne? You touched on the biological foundation of that, and thank you for doing this, Jared. Now, there is a question here related to mitochondria. Dr. Younger, do you think that the mitochondria are involved in this disease, and if so, how? Again, that's a very broad question, but I appreciate if you touch on this succinctly.
Yeah, and unfortunately that's one area that I'm not well qualified to talk about. That is a completely separate part of biology from what I'm trained in and what I'm really good at researching. So I really hope that that angle, because it is a compelling hypothesis, and I hope and I do think that other researchers are pursuing that. In fact, I know that's the case, and there's more stuff being done with the mitochondria than you've probably heard about. So there's people working on it, and I think I'm going to a meeting in a couple of months where that's going to be a big focus on getting some funding for people to look at that angle. So kind of thinking about how is the energy produced for the body and how is energy produced by the cells. You have to get down to these really small mitochondria, and that is a potential place for dysregulation. So kind of as scientists, we all pick our area. So I do neuroinflammation and immune stuff. Other people do mitochondria, and we see who's right. So it could be, it's very possible that I'll do these brain scans and I won't find anything, and the reason is because I should have been looking at mitochondria. But that's our role as scientists, is if I see it's a dead end, I got to say, okay, scrap it. What's the next best hypothesis? And that second may be mitochondria, and then we'll shift and look at that. But in the meantime, I don't know the literature well enough to actually explain how those can be involved and how much support it has in the scientific literature.
Thank you for this explanation. Certainly signaling pathways are extrapolatable in many ways, and we have strong programs here at Solve MECFS on the bioenergetics and the mitochondrial biology. So we published in our recent newsletter and print publication a summary of this, but this is your presentation. That's why I'm posing the questions as they come, and definitely I echo your call to follow this exciting area of biology. Now, another question. In your opinion, does brain inflammation affect fibromyalgia sufferers? This is a fibromyalgia question.
Yeah, I think so. The big debate now is whether MECFS and fibromyalgia are related disorders, the same disorder, or completely separate. And I know that's very controversial. Even inside the science world, there's a lot of controversy. I've been doing this kind of stuff for over 10 years. Right now, I'm working from the hypothesis that fibromyalgia and CFS mostly involve the same mechanisms, and I think that if we find an advancement in one, it's going to help us on the other. So I tend to combine those two. Of course, I'm keeping my mind open to the fact that they're different, but basically all the things I just described, we're also going to do that in fibromyalgia. A lot of people fit the criteria for both, so most people with fibromyalgia also meet chronic fatigue syndrome criteria. Now, the reverse isn't necessarily true. There's a lot of people with CFS that don't have chronic pain, but there is a lot of overlap. So we'll look at people with CFS, we'll look at people with fibromyalgia, and we'll look at people with both, and to make sure to see whether those are the same thing or not. But definitely everything I just talked about I think is applicable to fibromyalgia.
Fantastic. There are so many questions still, Jared, and I hope that you can work with us after this to answer them. We can post them later on our website. Is that something you can do?
Yeah, absolutely.
I really do not want to keep our friends and listeners longer than 1 hour. We promised them only 1 hour, although we must get you back at some point because there are so many exciting things, and please keep us up to date on the result of the immune tracking that you are doing. What you are doing is fascinating, and we are behind you. You know that our organization support brilliant people like you, and this is your organization, so we look forward to hearing more from you. And I would like to thank everyone who is on this call, and stay tuned to our next webinar. Thank you so much.
All right, thank you.